Hello there. So in our previous video, we had a look of the anatomy of plasma membrane. We discussed a little bit of the history of how we came up with the current understanding of the model of its structure, and we had a look of the fluid mosaic model of it. In addition, we studied phospholipid structure and how this gave a rise to the hydrophilic head, part, and hydrophobic tails. And this gave us the basis of understanding the spontaneous nature of forming a phospholipid bilayers. And we wrapped up the video by looking at the proteins. So now it's time for us to move our focus on the physiology of the plasma membranes. So our focus will be on this video mainly on the function, how and why they work the way that they do. One of the things that I mentioned at the very beginning of our first video was this, selective permeability. And this term describes the function of plasma membranes really well. They let some materials through it while blocking others from passing through them. And this is vital for forming the differences between the two sides of the cell membranes. In fact, this is the foundation of entire life, the very topic that we are studying on this course. The other topic that I want to discuss in this context is the two large categories of how materials may cross the cell membrane. And these are active and passive transport. Let's have a look at the latter first. So passive transport is something that we can consider as happening naturally. This does not require any energy investment. It just happens by itself because that's just a normal thing to happen. And usually this is down to a concentration gradient. So let's remind ourselves of what this was all about. Here we have a bottle of water to which we add a little food coloring on the corner. And of course, this means that the concentration of the food coloring is high in that corner, while it is low everywhere else in the bottle, right? And we remember that these concentrations always aim to reach a state of balance, where both sides have an equal amount of material. So an equilibrium, which is the term that some of you might have heard about this being used. So over time, these concentration gradients even out so that there is the same amount of food coloring in all parts of the bottle, right? So this is the basic concept of the concentration gradients. Materials always travel from areas of high concentration to areas of low concentration until these are equal. And this happens passively without any extra energy being invested into this event. And this is exactly what happens naturally in passive transport through plasma membrane. If the materials are small enough and allowed to pass through the membrane, they keep passing through it until the concentration on both sides is equal. So that is our passive transport. No energy needed for this to happen. It just happens naturally in nature. But in case of active transport, the situation is quite different. Here, energy is spent to move materials through the plasma membrane. And often it is that we are moving materials against their natural concentration gradient. So to use our previous example of the food coloring added to the bottle of water, we are moving these materials from areas of low concentration to areas of high concentration. So this is against concentration gradient. In other words, against what would normally happen. And of course, because this is against what would normally happen, this will require energy from us. Remember, that is what the green arrow that we saw earlier, from low to high. And establishing this buildup of different amounts of materials on different sides of the plasma membrane against their natural gradient requires energy. So, the big take-home message from this section is that some materials cross plasma membrane passively, which means that there is no need for energy for making this happen. They move along their normal concentration gradient. While active transport, these materials cross plasma membrane to the opposite direction of where they would normally go, and this is going to need energy to make it happen. And there are different kinds of active and passive transport methods. Let's have a look at some of these. Let's have a look at diffusion as our first example. And this is going to be one that falls under the category of passive transport. So in diffusion, there is initially an uneven concentration of substances on one side of the membrane in comparison to the other side. So this gives us a high concentration on one side and a low concentration on other side. And what we eventually want to do is to have the concentrations even on both sides. This is accomplished in diffusion by the plasma membrane becoming permeable for this material, and the materials moving from an area of high concentration to the areas of low concentration. And this continues until the concentrations are the same on both sides. Note that it is only the substances that were uneven in the beginning that move through this membrane. So this is what happens in diffusion. I think that our discussion is a concept that most students grasp really well almost right away. But let's think of a problem that we can have sometimes. What if this material, these red dots, would be unable to cross this membrane, but we still want to get the same concentration on these both sides? That's a good problem, isn't it? Well, in this case, we would be looking at osmosis. So here, the starting situation is similar. There is more substances on one side than on the other side. And this is, of course, another example of passive transport. As we mentioned earlier, now the substances that we have in different concentrations, so these red balls, they cannot cross the plasma membrane. So they might be physically too large, or the membrane does not let that happen for some other reason. But we still want to get the same concentration on both sides, right? Well, to get the same concentration established on both sides, the only way we can accomplish this is here by, rather than these red substances moving, is by having water moving from one side to another. And this water moves until we have the same concentration. Notice how water moved from an area of low concentration to the areas of high concentration until the concentration of this substance, symbolized by the red dots, is the same on both sides. So this is how osmosis, our second example of passive transport, worked. Notice that unlike in diffusion, where the substance itself moved, in osmosis, it is the water that moves to balance concentrations on both sides of the plasma membrane. Well, if you're like anything that I was as a student, at this point you're probably thinking, well, that is all very nice and good, but how does this apply to anything in real life that I would need to know it? Well, it does. And the example that we will use is tonicity of cells. This is relevant in many contexts, as we will see shortly, but the one that I have picked here is its relation to the fluid therapy, where we give fluids intravenously to the patient in order to compensate for fluid loss in their body. This is quite common. You often see patients with a drip in hospitals. And in fact, it is one of the first things that we often end up doing. We will talk about tonicity here, but before we dive deeper into the content, I want to remind you, although I'm pretty sure that you remember this, that in diffusion, we always had substances moving from areas of high concentration to areas of low concentration. And the goal was to even out the differences, right? And the example that we used earlier was this. A little food coloring dropped to the one corner of the glass of water, which would naturally result in high concentration of the coloring being in the left corner, and the rest of the glass having low concentration of it, right? And we saw that soon enough, this food color did spread to all parts of the glass until the concentrations were the same. This is the basis that all of our discussion on tonicity will build upon. Remember that we always try to even out this difference by moving the substances. And the first example that we will have a look is going to be hypotonic cell. In these cells, inside the cell has more charged salts or anything else that we may be studying. But let's just go with that as an example. So the inside of the cell is very salty. And these salts cannot cross the membranes in this example. So it is going to be all about osmosis that takes place. And what your body will try to do is that water will be flowing into the cell. The goal is that by adding more water, the contents inside the cell will become more diluted. In fact, to the same concentration as the outside of the cell has. Well, as a result, the cell will gain water and it will swell as more water flows in. And if the difference in concentrations is too high, the water will just keep flowing in more and more, and as a result, the cell may even end up to burst. This would not be good, would it? How are you going to remember this? Well, in biology, like in all other subjects, it is a good idea to always relate things to something that you can easily remember. So when I hear hypotonic cells, I think of a hippo. These are also big and bloated, just like these cells. So hypotonic cells are in my mind hippo cells, which are big and swollen so that water is flowing into these cells. Okay, next let's have a look of isotonic cells. In isotonic cells, the amount of salts is the same on both sides of the cell membrane. And as a result, the net movement of water is in total 0. So while some water may come into the cell, same amount of water also leaves the cell. So there is not in total any change in the water amount really. So as a consequence, the size of the cell remains the same too. Well, how can you remember this? Well, to me, when I hear isotonic, I think of this creature that says, I'm so perfect. So if you are perfect, you do not need to change. That is because you are perfect as you are. So for me, isotonic cells are isoperfect cells. They are perfect, so they remain the same. Okay, let's have a look at the final one. This will be hypertonic cells. In hypertonic cells, the inside of the cell is less salty than the outside. So in other words, the outside is more salty. What does this cause to happen? As a result, water will flow out from the cell, trying to dilute the cell concentration outside it. And this will keep happening, causing the cell to shrink. And if the difference is in concentrations is too big, the cell may eventually even end up drying up. So how do I remember this? Well, when I think of hypertonic cells, I think of what kind of a person is a person that is hyper. Well, these persons have a lot of energy, right? And people who have a lot of energy, what do? Well, they run around a lot. So what happens if you run around a lot? You become skinny. So for me, hypertonic cells are skinny cells. So they lose fluids and shrink in size. So that was our review of the tonicity. I hope that this and the mnemonic tools that I provided helped. If you would like to have another review, you can check out a video that I have linked to the very end of this video, and that video revives the same concept in my classroom as a lecture clip. So I hope that that helps. So where can we see this in nature? Well, the plants also show this. We do not want to water plant with salty water, because this would only lead to it drying up even more. But if we water the plant with water that is not salty, see. But of course, I would imagine that most of you are not necessarily heading to a plant science field. How about human health instead? And here I have a rather interesting question for you. Can you die out of drinking too much water? And the answer is yes, it is possible. If you consume a large amount of normal drinking water without any extra electrolytes in it, and you do not let your body to get rid of this extra fluid, for example by going to pee, then it is possible that this may even come fatal in extreme cases. And this is exactly what happened in one competition arranged by a local radio station. I think that the competition was named as Hold Your Wii for a Wii, or something like that. Remember when the Wii gaming consoles first came to the market? This was at that time. So this one woman did just that, and the outcome was devastating. So which of the tonicity conditions that we just reviewed would you apply here? And this would be, of course, an example of the cells becoming hypotonic, because there is this extra water around them. And your body will try to balance off this salt difference with the insides of the cells being relatively a little saltier than the outsides. So fluids, because of this was just pure drinking water, with nothing added to it to make it more balanced, well, now fluids will flow into the cells. How sad. I guess that the moral of the story is that everyone, including radio station DJs, should study some basic biology. In fact, some simple sea animals living in water have to be careful about this too. Here we see a bright field light microscopy image at 480 times magnification of a Paramecium. They have to constantly pump water out of their body to keep it from bursting in a hypotonic medium. So let's have a look at one more example of passive transport. And this is called facilitated transport. And as we start to consider this transport mechanism, please remember the process of diffusion, which was also passive. So always from high concentration to low concentration. But this time there is a problem. These molecules or ions symbolized by the red dots, they cannot pass across the membrane. There may be many reasons for this, for example, their size, charges, or something else. So we have to think of another way to do this. And this is by recruiting some integral proteins that are specific for this function. Remember that we called these integral because they extended through the entire thickness of the membrane, while peripheral proteins did not. So back to this situation where we were. And now, thanks to these proteins that help with this, we are able to have these substances symbolized by the red dots to travel across the membrane. And this continues until the concentrations are same on both sides. There's one more thing that I want to mention about these proteins involved here. These integral proteins that help, they can be either channel proteins, which are simply described just like a tube that allows materials to pass through, or they can be carrier proteins, which then change their own shape in order to physically carry the substance across the membrane. So that was the last of our passive transport. And now I would like us to look at a little bit of the active transport. And the first one that we will look at is going to be different kinds of protein pumps on the membrane. And they do just what you would think the pumps would do. They move something across, but to do so, additional energy is needed. And let's have a look at this with an example. And in this example, colors are important. You can see that the red dots carry a positive charge, while the blue ones carry a negative charge. And you also see that there is as many of these on the both sides. So this is all in balance, just like we wanted it to, right? Well, sometimes we do not want this balanced state. Instead, we might want to establish a charge difference so that in the end, one side is more negative while the other side is more positive. And because this is against of what would naturally happen, this requires energy. So there can be one or many pumps, and eventually we have this difference in charges inside the cell and outside the cell. Well, you might wonder, when can this be useful for us? Let's consider one example. Where we might see an example of this is with neurons, which are our nerve cells. And nerve cells carry messages from one part of the body to another. And these messages are carried as electrical impulses, right? And to do this, we need to first have a negative charge inside the nerve cell. And this is at its resting state. And when an electrical impulse travels, this changes momentarily into positive. In fact, it is negative 70 millivolts that the charge inside the nerve cell typically is. Well, the final concept for this video will be one more example of active transport. In fact, it uses a lot of energy. And now we have a situation where we either have a lot of material or material is so big that it cannot physically go through the plasma membrane or proteins. And this is known as bulk transport. So what are we going to do to deal with this? Well, we have two possible ways. In endocytosis, the material that we are looking at, remember there was a lot of it, or it was too large to pass. Well, this material is taken into the cell. So what happens is that the plasma membrane forms a sac around this material, and then this sac can be engulfed as it fuses with the cell wall, and then once inside may continue transporting these materials as if it was a vesicle. Whereas during exocytosis, we move this material out from the cell. So one way to think of these two types of bulk transport is to imagine that endocytosis, the cell eats something in. I do not know, endo can stand for entry, for example, if that helps you to remember. And exocytosis, instead, it's almost like the cell would vomit something out from it, I guess. You can rhyme in your mind the exo with exit. I hope that helps. And now we have reached the end of this chapter. On the first video, we looked at the structure of plasma membranes. And on this one, we have focused on the actions related to it. I hope that you have found this helpful. I will see you in the lab.